Okay, so I want you to close your eyes and just imagine the perfect cup of coffee. And I don't mean just a good cup. I mean like the absolute pinnacle of what coffee can be. Forget the cheap stuff, forget the daily grind. This is an experience. This coffee is. It's silken smooth, almost buttery or syrupy in its texture. It hits your tongue with these incredible deep notes. Dark chocolate, maybe vanilla. Sometimes you even get a hint of mint or spice.
Yeah.
But the key thing, the absolute hallmark, is the finish. It's unbelievably clean. There's almost no lingering bitterness, and the aroma is just complex and arresting.
That description alone sounds expensive. Like $50 expensive. That sounds like something you'd find behind a velvet rope.
That's it.
Where the story of how it's made is almost as important as the taste. This is the stuff of legend, right? The kind of brew people fly halfway across the world for.
Exactly. We are talking about the mythical brew known as Kopi Luwak, or civet coffee. And this is a legitimate luxury item. Depending on where you buy it and whether you're getting the real deal, it can cost anywhere from $30 to upwards of $80 for a single cup.
$80.
For one cup. It has built this intense reputation, this almost spiritual aura around its unique, exceptionally low bitterness profile.
And, okay, here's where the story takes a very sharp left turn into the bizarre. Because we know where those beautiful, aromatic, low bitterness beans come from. And you just can't talk about Kopi Luwak without talking about its deeply unusual and, frankly, kind of gross origin.
It's the moment of shock, if you haven't heard it before. This incredibly expensive coffee is made from beans that have passed entirely through the digestive tract of a small nocturnal mammal native to Asia, the Asian Palm Civet.
Wow.
The civet eats the cherry, it digests the sweet outer pulp, and the hard inner seed, the bean itself, is excreted whole. That's the raw material for this multi-million-dollar industry.
Dude, Kopi Luwak, it literally translates to civet coffee.
Yeah.
And it's the kind of thing you hear and your immediate, your natural skepticism just kicks in. This has to be pure hype. It's a spectacular, bizarre story that's just used to justify an obscene price tag for what is, essentially processed animal waste.
And that initial gut-level skepticism is precisely what drives this deep dive. We're moving past the U factor, past the shock value, and we're diving straight into the chemistry. We need to answer the core question posed by you, the listener. Is the difference purely hype and spectacle, or does the civet gut fundamentally change the bean's chemistry in a way that is scientifically measurable?
Yeah. Can we actually put a chemical number on that smoothness, on that low bitterness?
Exactly.
Because if the civet's only contribution is that it, selects the ripest, highest quality fruit, which a person could do, then the price makes absolutely no sense.
It would be meaningless.
We have to know if the actual messy biological process is the secret sauce. Does the animal's stomach acid and its microbiome turn a good bean into a great, unique bean?
Okay, let's unpack this incredible, bizarre journey and see what happens inside that little animal. So the whole process starts with the star of our show, the civet. Specifically, the common Asian palm civet, scientifically known as Paradoxurus hermaphroditus.
Right.
These are slender, dark, masked, almost cat-like creatures found across large parts of South and Southeast Asia, everywhere from Indonesia to the Philippines, down through Vietnam, and even in these biodiversity hotspots like the Western Ghats in India.
I'm picturing them as these sort of nocturnal, slightly mischievous quality control agents of the forest. And their role isn't passive, right? This isn't just an animal stumbling upon a coffee plant.
No, not at all. Their consumption of coffee is highly deliberate, especially during the dry season when other ripe fruits might be scarce. Think of the civet as the initial, most selective quality controller in the entire world. They don't just eat any old coffee cherry. They rely on their highly refined, instinctual palate to select only the ripest, most flawless fruit.
So that selective harvesting is really the first layer of premium quality.
It is.
Because in traditional coffee processing, you always get faulty or underripe cherries. They call them floaters. And those can introduce sour or bitter flavors to the final batch. The civet, by only choosing the perfect ones, just eliminates that risk from the get-go. They're basically pre-sorting for us.
Exactly. This initial selection is vital. It guarantees the highest possible starting quality. But, like we said, if this were the only difference, it wouldn't justify the price. The real magic, the part that required the deep chemical dive, happens once the bean enters what researchers very dryly refer to as the transformation chamber.
Which is the civet's gastrointestinal tract.
The civet's gut.
So this is where we stop talking about agriculture and we start talking about biochemistry.
100%. The coffee seed, the bean, is incredibly tough and hard, so it passes through the system relatively intact. The surrounding soft pulp gets fully digested. But while it's in transit, which is typically 24 to 36 hours, it's subjected to what researchers call digestive bioprocessing. And this is a spontaneous, high-efficiency fermentation method, unlike anything you could cleanly or easily replicate in a coffee mill.
Okay, so this process basically replaces the traditional fermentation step, where coffee processors soak the beans to remove that sticky mucilage and kickstart all the enzymes. But instead of just a simple soak in water, this is an internal chemical enzyme high-pressure wash.
You've got three major chemical forces at work here, all acting together in a really unique way. First, you have the intense acidic treatment from the gastric juices, which partially breaks down the outer protective layers of the bean. Second, you have powerful enzymatic action. Digestive enzymes like pepsin, which breaks down proteins and amylases and proteases, they permeate the endocarp. That's the tough outer casing of the coffee seed.
And that's the key challenge, isn't it? Getting the change into the bean. The bean doesn't want to break down. It's designed by nature to pass through, germinate, and survive.
Right.
So the enzymes have to be aggressive enough to actually penetrate those tough outer layers.
They are. And the third, and maybe the most mysterious force, is the microbial fermentation, which is provided by the unique gut flora, or the microbiome, of the civet.
So it's not just an acid wash. This is a highly specialized chemical and biological bath for the coffee bean.
Precisely. In the coffee world, fermentation has two main goals. Clear the bean of all that mucilage and start the flavor development. The civet gut does this job, but because it happens internally and under high temperature conditions, the animal's body temperature, the chemical action is incredibly precise and unique. And one of the immediate crucial chemical impacts that the source has documented is the breakdown of storage proteins. Coffee beans store flavor precursors as these long-chain proteins. This digestive process breaks those long chains down into shorter chains, shorter peptides, and that fundamentally alters the bean's amino acid composition.
Wow. Okay, that is a massive finding. Proteins and amino acids, those are the fuel for the Maillard reaction later on, right?
Exactly.
They're the compounds that create all the wonderful complex aromas and flavors when you actually roast the bean. So if you change the raw ingredients, you're going to change the final dish dramatically. Changing the protein structure is a direct path to altering the final flavor.
Exactly. And the most immediate flavor impact of this protein modification is a reduction in bitterness. Many bitter compounds in coffee are linked to these longer chain proteins. By cleaving them into smaller peptides, the civet is essentially pre-digesting the bitterness out of the bean before it even gets near a roaster.
I think I remember the sources also mentioning that the digestive process initiates germination, which is sometimes called malting.
That's right. Malting, which is often done deliberately in grains for brewing, is known to significantly reduce bitterness and change starch content. So the civet is essentially pre-malting the bean for us internally. Think of the civet as replacing two traditionally distinct complex steps, selective hand-picking and wet fermentation processing, with a single, integrated, high-efficiency biological method.
And that chemically explains the low bitterness profile that makes Kopi Luwak so legendary. It's not magic, it's just enzymatic chemistry.
It is a natural factory. The finished product, once it's collected, cleaned, dried, and roasted, should theoretically be a superior, less bitter, and uniquely flavored bean.
But this is where we meet the villain in our story, the huge stakes of authenticity. If the finished product is worth $80 a cup, and the production method involves foraging through animal droppings, which is inherently rare and inconsistent, the market is just absolutely ripe for fraud.
And the sources confirm this has been a catastrophic problem for the industry. Because Kopi Luwak is rare, valuable, and involves such a messy process, counterfeiting is a massive, massive industry problem. If you can take cheap, regular coffee, just slap a Kopi Luwak label on it, and sell it for a gigantic markup, a lot of people will.
And this isn't just some minor marketing issue. This fraud undermines consumer trust, it makes ethical sourcing impossible, and it just confuses the entire market. So the problem of fraud wasn't solved by better farming, it had to be solved by better science.
Precisely. This severe fraud problem is what necessitated the shift in research. Scientists had to become chemical detectives. They needed a bulletproof way to differentiate a real civet-processed bean from a fake, manually processed bean. The question shifted from, does it taste good, to the much more objective and measurable question, is it real, and how do we prove it chemically? And this pushed the need for forensic chemical profiling, or metabolomics, which launches us right into the scientific quest. To solve the fraud problem and to really understand the core science, researchers had to employ a serious scientific arsenal. I mean, we're talking about technologies that can detect these minute changes in molecular structure and concentration, often measured in parts per million.
Okay, so we've officially left the jungle floor and we've arrived in the sterile, high-tech lab. What kind of tools were they wielding? What were they specifically looking for?
Well, they needed tools that could analyze two different types of chemical compounds. First, the volatile compounds, the things that vaporize easily and contribute to the aroma. And second, the non-volatile compounds, the bigger, heavier molecules like fats, proteins, and minerals. So for the volatile components, they relied heavily on techniques like gas chromatography, coupled with mass spectrometry, or GCMS. They also used GC coupled with a flame ionization detector, GCFID, for more rapid industry screening.
Okay, let's break down those acronyms. If we're using our analogy engine, how do we explain something like GCMS?
Let's stick with the food analogy. If coffee flavor is a finished recipe, let's say it's a complex cake, GCMS is the tool that reads the smell of the ingredients. It takes all the aroma components, the tiny vapor molecules, and it separates them out one by one. That's the chromatography part. And then it weighs each one precisely. That's the mass spectrometry part. It tells you exactly what kind of butter, what specific spices, and what proportion of vanilla were in that mix.
So GCMS handles the delicate aromatic evidence left by the civet. It identifies the chemical signature of the smell.
That's half the picture. For the non-volatile compounds, the fats and proteins, they used instruments like ultra-performance liquid chromatography with mass spectrometry, UPLC-MS, and also nuclear magnetic resonance, or NMR, spectroscopy. These tools handle the heavier structural ingredients.
So they weren't just tasting the cake, they were reading the entire detailed blueprint of the kitchen, identifying every single ingredient in its exact quantity.
That is the essence of metabolomics, identifying and measuring every single one of the volatile and non-volatile compounds, the metabolites, that contribute to the bean's chemical makeup. The key was comparison. They rigorously compared unroasted, civet-collected beans, and they specifically used both Arabica and Robusta varieties with manually harvested control beans.
Why unroasted? Why is that detail so important?
Oh, it's critical. They specifically avoided deep roasting at this investigative stage because roasting itself drastically alters the chemistry. If you apply 400 degrees Fahrenheit of heat, you create thousands of new molecules from the Maillard reaction. Researchers wanted the native, raw chemical profile left only by the civet's digestive action. They needed the pure signature.
That's smart. If roasting introduces too much noise, you can't isolate the civet's specific chemical signature.
And once they collected this massive amount of chemical data, I mean, sometimes profiling over 37 different compounds, they couldn't just read it off a spreadsheet. They needed statistical models to find the patterns. So they used these statistical detectives called chemometrics, like principal component analysis, or PCA, and OPLS-DA.
Wait, how does a statistical model become a detective?
Okay, think of it like this. Imagine you have 100 suspects, which are your coffee samples, and you have 37 characteristics, the metabolites, for each one. PCA doesn't know who the fakers are, but it looks at all 100 suspects and says, based on these 37 characteristics, these 10 samples are mathematically clustered way over here, and these other 90 samples are clustered over there. The statistical model just groups the samples based on subtle differences in their composition. It doesn't care what the label says, it just looks at the chemical evidence.
That's fascinating. So what did the data tell the detectives? Where did the authentic civet coffee group diverge most significantly from the manually harvested control coffee?
The core discovery centered around the lipid profile, the fats. They found statistically significant markers in the civet coffee that are the chemical fingerprint of that digestion process.
Okay.
First, total fat content was significantly higher in civet beans. And this wasn't a slight difference. For instance, in one detailed study focusing on Robusta beans, the civet-processed beans averaged 8.364% total fat compared to 5.946% in the manually processed beans.
That is a staggering difference. That's like a 40% increase in total fat content. Why does that matter in the cup?
Higher fat content translates directly to the perceived smoothness and body in the final brew. Fat molecules trap and suspend aroma compounds, and they coat the tongue, creating that desirable, syrupy or silken mouthfeel that Kopi Luwak is famous for. This high-fat content is the chemical explanation for the smoothness.
So we can literally put a number on the smoothness now. It's the 8.364% total fat content.
But it gets even more specific than total fat. They found unique molecular markers called fatty acid methyl esters, or FAMEs. Specifically, caprylic acid methyl ester and capric acid methyl ester were significantly elevated in the scat-derived beans. And when I say significantly, the statistical probability that this occurred randomly, the p-value, was incredibly low. This was a reliable chemical change caused by the civet's digestion.
Caprylic and capric acids, are these just generic fats or do they contribute specific flavors?
They are crucial flavor precursors, but they also have specific sensory contributions. Caprylic acid, or C8, and capric acid, C10, are medium-chain fatty acids. In high concentrations, they can sometimes carry sharp, slightly acidic, or waxy notes. But in controlled lower concentrations, they contribute to the overall complexity and smoothness. Their elevated presence suggests why Kopi Luwak has its unique aroma. They're acting as enhanced flavor carriers.
And this elevation is the so what? I mean, the animal's digestive process isn't just dissolving the pulp, it's chemically enriching the bean core with specific desirable fatty acids that enhance the mouthfeel and aroma complexity. But how does that happen? Is it just the acid in the stomach, or is it the living elements, the microbes?
That brings us to the indispensable microbial engine. The fermentation step relies heavily on bacteria and yeast. Researchers thoroughly investigated the civet's gut microbiome and identified specific microorganisms involved, most notably the Gluconobacter species.
Gluconobacter. I recognize that name from traditional fermentation. Isn't that what's often involved in making vinegar, or even in the early stages of cocoa fermentation?
Exactly. They're acetic acid bacteria. They're linked directly to the fermentation process that drives flavor development in the civet's digestive tract. These bacteria are responsible for breaking down complex molecules, potentially leading to the formation of those elevated flames we were just talking about.
That's a perfect analogy then. Traditional fermentation relies on natural yeasts or sometimes specific starter cultures like a specialized sourdough starter. But the civet gut is like a specially selected, perfectly balanced instant yeast packet, a high-performance internal microbiome that acts on the coffee bean from the inside out. And it promotes the specific chemical reactions that lead to those elevated fatty acid methyl esters and the overall higher fat content. The civet isn't just a transport system, it's an active biological refinery.
And this microbial signature changes the whole game, chemically speaking. It provides a distinct, measurable difference that can be used for forensic authentication. We have moved from speculation to provable facts based on specific molecular concentrations. So we've established that the civet's digestive bioprocessing chemically alters the flavor profile. It increases key fatty acids, total fat content, and reduces bitterness. But the research didn't stop at flavor. They started looking at nutritional and functional compounds. They shifted the focus to whether Kopi Luwak could be considered a functional food, a food that offers health benefits beyond just basic nutrition.
Wait, hold on. You're telling me this bizarre exotic coffee might actually be good for you from a clinical perspective? That would be the ultimate, unexpected justification for the astronomical price.
It's a compelling finding, and it's thanks to mineral and antioxidant analysis performed using high-tech tools like ICP-MS, that's inductively coupled plasma mass spectrometry, which analyzes elemental composition. Kopi Luwak demonstrated significantly higher levels of total polyphenols and consequently strong antioxidant activity compared to regular coffee.
Wow. Polyphenols are the compounds often associated with the health benefits in things like red wine, green tea, and cocoa, right? They're the molecular heavy lifters fighting oxidative stress. How significant was this boost?
The comparative difference was striking. When they tested it using the DPPH radical scavenging assay, which is a standard test for antioxidant capacity, the IC50 value for Kopi Luwak was measured as significantly lower. It was 61.4 milligrams per deciliter.
Compared to what?
Compared to 93.6 milligrams per deciliter for regular coffee.
Okay, for clarity, a lower IC50 means you need less the substance to get the same level of antioxidant action. So less of the civet coffee sample was required to neutralize the free radicals in the test. That is a huge indicator that something fundamental changed during the process.
Absolutely. The potency was clearly enhanced, and the mechanism is the real marvel. Researchers hypothesized that the specialized magic of the civet's gut microbiota is the key. When the coffee cherry passes through, the gut microbiota releases non-extractable bound polyphenols from the coffee cherry material.
Non-extractable bound polyphenols. So these are beneficial compounds that are usually locked away, chemically bound to the fiber and structural material of the bean, and our normal brewing methods just can't access them.
Exactly. They're like treasure locked in a safe. But the civet's intense enzymatic action and microbial fermentation, that powerful biological processing, it acts like the key. It makes those beneficial compounds more bioavailable and increases the final product's total antioxidant potency.
This finding elevates the entire discussion. We're not just talking about a smoother taste anymore. This implies that Kopi Luwak, if you source it ethically, could genuinely be considered an important candidate for functional foods, something far beyond just a novelty beverage.
And beyond the organic compounds, there were interesting mineral shifts too, although the sources suggest these elemental changes might not affect the taste directly. Using the ICP-MS system to analyze 23 different trace minerals, researchers confirmed that the civet's digestive process alters the bean's elemental composition.
What specifically changed in the mineral scorecard?
Civet coffee showed lower concentrations of several trace minerals, specifically manganese, barium, chromium, and zinc. This decrease is likely due to the highly acidic environment of the stomach and the digestive absorption processes. The animal itself is pulling some of these micronutrients out for its own use as the bean passes through.
And what about increases?
Conversely, the civet coffee showed higher concentrations of copper and rubidium compared to regular coffee. This could be due to the slight absorption of these elements from the civet's diet or digestive tract itself, adhering to the outer surface of the bean. And while these minor elemental changes probably don't affect palatability, they serve as yet another unique chemical signature of the digestive bioprocessing.
So we have elevated fats for smoothness, protein modification for lower bitterness, and now chemically increased antioxidant power. Plus, the mineral shifts provide a secondary layer of proof. What about the basic nutritional factors we usually worry about, like caffeine?
Well, other studies corroborated earlier anecdotal evidence that civet coffee typically contains slightly lower caffeine and acidity bitterness, a highly desired trait for premium markets. One specific study focusing on an in vitro process that mimicked the civet's gut using fecal suspensions showed they could reduce caffeine content from 2.2% to 1.8% within 24 hours. They also degraded about 84% of the sugar content during that time.
So that lower caffeine, lower acidity, lower bitterness profile, combined with the higher fat content for smoothness, that is the complete picture that explains the unique premium taste experience. The science absolutely backs up the legend.
Now let's circle back to the villain of fraud. Since taste is subjective and counterfeits are rampant, the entire purpose of this deep dive into chemistry was to create objective, measurable criteria for authentication.
Right. If the difference is provable through chemistry, like those unique elevated fatty acid methyl esters, then we can definitively shut down the fakers, regardless of how good their knockoff coffee tastes.
That's the game changer. The robust chemical differences are used to distinguish the real deal from the fake. The future of authentication lies in Enzymatic and these rapid, cost-effective technologies that move beyond just laboratory testing. This involves using metabolite fingerprinting via systems like GCFID, which provides a fast and reliable analysis for industry screening.
So instead of sending a sample off to a lab for 2 weeks, they want something that can give them an answer much faster, maybe right on site.
Exactly. And the most futuristic application involves integrating machine learning algorithms with sensory tools. Researchers are using electronic nose, or e-nose, systems. These machines essentially smell the coffee's molecular profile and convert that into digital data. This data is then fed into machine learning algorithms like support vector machine, or SVM, and random forest.
So we have a smart machine tasting the coffee based on its molecular signature. That's incredible, like a robot sommelier.
That's basically it. And these systems achieved extraordinary accuracy, sometimes 90 to 100% in differentiating authentic civet coffee based purely on the aroma fingerprint. The computer can detect the subtle presence of those telltale capric acid methyl esters and other unique volatile compounds faster and more reliably than a human taster or even a standard lab test.
So not only have scientists proven the chemical difference, they've armed industry regulators with the tools to protect consumer trust and fight against counterfeiting. It means that if you pay that astronomical price, the chemistry better back up the claim, or the e-nose is going to rat you out.
Okay, so now we have a definitive chemical scorecard. Kopi Luwak is scientifically distinct. We're talking elevated fats, specific methyl esters like capric and caprylic acid, a reduction in bitterness and caffeine, and surprisingly, an enhancement in antioxidant capacity and polyphenols. The unique taste is rooted in provable, quantifiable chemistry.
The hype is backed by data. The unique taste is real, and the mechanism is this internal fermentation and enzymatic modification. But the scientific victory really just highlights the deeper challenge facing this entire industry, the ethical imperative. The moment the demand for Kopi Luwak exceeded the unpredictable supply you could find in the wild, it created a perverse economic incentive.
And that's the critical discussion. We have to acknowledge that persistent ethical concerns regarding civet captivity and animal welfare, which fundamentally undermine any benefit derived from the product. The civet farming industry, where animals are caged, often in terrible conditions, and force-fed coffee, is cruel and unsustainable.
It completely defeats the purpose. The initial quality relies on the civets selectively choosing only the perfect, ripest cherries in the wild. When they're caged and force-fed, that crucial, high-quality selection step is completely lost. So the starting material is already compromised. You get the scat, but you don't get the quality.
Right. And while some areas still rely on truly wild civets, where they aid in seed dispersal and defecate in specific, elevated spots that are easy for collectors to find, the commercial reality is dominated by harmful captive farming. The future viability of this industry, if it even exists at all, hinges entirely on ethical sourcing, transparency, and certification frameworks that prove the civets were not caged.
So if the animal is the perfect yet highly problematic bioreactor, and using the animal is unethical and actually lowers the quality, how do we get the chemical benefits without the cruelty? The science we just detailed should provide the path to skip the animal entirely.
And this is where the biotechnological alternatives step in. Since we know what chemicals change, the precise enzymes involved, the fat profile, the microbial input, we can attempt to replicate the civet gut in a controlled, industrialized laboratory environment. This is the new frontier, biotransformed coffee.
Synthesizing the civet gut environment in a tank. So tell me about the in vitro model development.
Well, researchers have focused on replicating the core digestive mechanisms. They found that treating coffee with an acidic solution to mimic gastric acid and key digestive enzymes like pepsin and proteases for a controlled duration, usually between 90 minutes and 24 hours, can successfully produce an enhanced aroma that is chemically similar to authentic civet coffee.
That time window, 90 minutes to 24 hours, that's a direct insight from the civet's digestive cycle. They don't need a month of fermentation, they just need a high-impact, short-duration chemical bath.
And the research goal now is combining that enzymatic bath with the missing link, the microbiome. They're working on using controlled microbial cultures in bioreactors to achieve that specific metabolic transformation without any animal involvement. We already know that microbes like Gluconobacter are key players, and other studies have investigated using specific safe bacteria like Bacillus subtilis or even mixed civet fecal cultures as highly specific starter cultures for fermentation.
If they can perfectly replicate the enzymatic and microbial fingerprint of the civet gut in a stainless steel tank, they remove the ethical problem entirely, and they stabilize the chemical output and eliminate the dependency on a small mammal's biology. It makes the process repeatable, scalable, and controllable.
It also solves the problem of quality, inconsistency, and fraud, because you control the entire input and output. The goal is to create biotransformed coffee that is chemical identical to, or perhaps even superior to, Kopi Luwak by isolating and industrializing the precise chemical instruction we've gleaned from the civet's digestion.
That is the truly ethical and sustainable future for this unique flavor profile. The animal was the discovery tool. The lab is the production factory.
That's the frontier. But before we sign off, there's one final critical provocative thought based on all this chemical discovery. Remember, much of the research we discussed focused on unroasted green beans, because the scientists wanted to measure the native chemical changes caused only by the civet.
Right. They established the unique raw ingredients that the civet produces, the high-fat content, the broken-down proteins, those specific FAMEs.
Exactly.
Yeah.
The civet creates specialized raw ingredients, higher fats, modified proteins, specific fatty acids. However, the final legendary flavor of Kopi Luwak, that amazing smoothness, that complex aroma, it doesn't happen until those unique biotransformed chemicals react to the intense heat of the roasting process. The chemical reactions that take place inside the coffee roaster are incredibly complex. We're talking about the Maillard reaction, the caramelization of residual sugars, and these reactions depend entirely on the precursors present in the green bean. If the civet has already broken down the proteins and boosted the caprylic and capric acids, the bean will react fundamentally differently to heat than a standard bean.
So the civet sets the stage by preparing the raw material, but the roaster delivers the final performance. If you roast a standard bean too dark, you often get a bitter, charred flavor. But what happens if you roast this high-fat, pre-digested, civet-engineered bean too aggressively?
You risk destroying the precise, valuable flavor precursors that the civet works so hard to create. Those specific caprylic and capric acid methyl esters are volatile. They can be easily stripped away or broken down further under intense heat. The unique qualities of Kopi Luwak exist in a very narrow, specialized roasting window.
So the next great chemical deep dive isn't the gut, it's the oven. It's about finding the perfect roast recipe optimized specifically for this unique biotransformed bean. The truly perfect $80 cup of Kopi Luwak isn't just about finding the right scat or building the right bioreactor. It's about scientifically calibrating the roasting profile to maximize the flavor potential of that unique molecular structure. The science doesn't end with the civet.
So what does this all mean? We've seen that Kopi Luwak is chemically distinct, proven by significantly elevated fats, unique volatile compounds influenced by Gluconobacter, and surprisingly high levels of polyphenols and antioxidants. The unique taste profile is absolutely rooted in provable chemistry.
The hype is backed by data. The unique taste is real, and the mechanism is internal fermentation and enzymatic modification.
So now, armed with this detailed chemical knowledge, you, the listener, get to decide not just what you drink, but how it was made, and whether the coming wave of biotechnology can replicate nature ethically and sustainably. We've gone from the jungle to the lab, all in the name of a better brew.
A fascinating and surprisingly complex adventure. Now, if you'll excuse me, I need to go look up a good Bacillus subtilis recipe.
We'll catch you on the next deep dive.